Again, welcome and good morning, my dear medical students, neuroscientists, physicians, and surgeons. As you all know, every person in this universe enters naked, but it is equipped with the brain, the part of central nervous system which controls the whole body. Body is controlled by the nervous system as well as the endocrine system, but master system which also control the endocrine system is the central nervous system. So how this central nervous system controls the body and how this central nervous system maintains the homeostasis of the body and how this system controls and protects the body from outer and internal milieu. As you all know, it is having a sensory system and a motor system. In my coming lectures, you will see how the cerebral cortex is having a primary motor cortex, premotor cortex and supplementary motor area, how the sensation or the stimuli of impulse of sensation reaches the primary motor area and how decision is making from the motor area and how the motor command is carried by the motor system and how the sensory system receives the impulse of different sensations. So central nervous system is having a sensory part and a motor part. So how we receive the sensation from the external environment as well as the internal milieu of the body. Human body is having some sensory nerve endings on its peripheral part, that is skin, largest part of the body, and also having receptors inside the body like blood vessels, heart, lungs and viscera of abdomen. Continuously round the clock, this central nervous system collects the information from external environment as well as the internal environment of the body. To make this possible, it is having some biological transducer. These biological transducers receives the stimuli which are in physical form. These receptors converts this physical energy in the electrical energy or impulse or action potential. So it is having receptors. So in simple words, I can say receptor is a biological transducer which convert the physical energy which may be in form of pain, touch, pressure, vibration or temperature in a action potential or in a form of electrical impulse. So what is the function of receptor? It converts the physical energy in the electrical energy. So it acts as a biological transducer. There are different types of receptors in human body. On the basis of the location of the receptor, there are three types of receptors. On the base of location of the receptor, the receptor may be exteroreceptor or ectoreceptor. The receptor which are exteroreceptor, maybe in the papilla, dermal papilla or dermis, or in the hairs of the skin. These may be receptors for pain, touch, temperature. Pain, touch, temperature, or receptors for hearing, vision, olfaction, taste and balance. So, reception for vision, hearing, that is auditory, balance, or olfaction, that is for smell and taste sensations. These are receptors which are for pain, touch, temperature, vision and hearing. Balance, olfaction and taste are located outside the body and known as exteroceptors. The other receptors known as proprioceptors. Proprioceptors are receptors located in the muscle, joint and tendon. These receptors receive the information about the contraction of the muscle, angulation of the joint and what is the orientation of the head in this 3D environment or 3D space. So, proprioceptors may be neuromuscular receptor, or it may be tendon organ, or in the joint it may be kinesthetic receptor, kinesthetic. So, proprioceptors are the receptors situated in the muscle, that is skeletal muscle, the joint and the tendon, and these receptors receive information about the degree of contraction of the muscle, what is the angulation of the joint and what is the position of the head in this 3D space and what is the initial position of the body in this 3D space. And third types of receptors which are inside the body known as interoceptors. These types of receptors are seen inside the blood vessels. Inside the heart, in the viscera of the abdomen. These receptors which are bringing the sensory information from the blood vessels, the GIT, the brain, the heart and the lungs. These stimuli or receptors are at the subconscious level. These sensations don't reach at the level of consciousness, and in case of stretch and severe pain in viscera, which is due to the stretching of the viscera, reach the level of consciousness. So, the stimuli received by interoceptors don't reach at the level of consciousness, and when the pain is severe due to stretching of the smooth muscles, reaches the level of consciousness. So, on the basis of location of the receptors, there are three types of receptors: exteroceptors, proprioceptors and interoceptors. On the basis of their microscopic structures, there are three types of receptors. microscopic. There are again three types or sometimes two types of receptors. The one is the receptors which are non-encapsulated. These receptors are not having a capsule of connective tissue outside these terminal sensory endings, which are modified in a form of receptor. So non-encapsulated receptors are non-encapsulated. There may be free nerve endings. There may be some hair, root hair plexus. These receptors, free nerve endings, are for pain and crude touch and temperature. Root hair plexus around the hairs of the skin, and the other receptors which are non-encapsulated are the Merkel's disc. Merkel's disc or Merkel's corpuscle, which is for the touch, and these receptors are seen in the palm and sole where hair are not seen. So the receptors which are not having a capsule of connective tissue, and three types of these non-encapsulated receptors: free nerve endings for pain and temperature, root hair plexus around the hair of the skin, and the non-hairy part of the body, that is the palm and sole, are having this type of receptor, that is Merkel's disc, for touch. The other receptors which are capsulated, there are different types of receptors which are capsulated and meant for the different types of stimuli. These capsulated receptors are Meissner's corpuscles, the Meissner's corpuscles for touch, and the Pacinian corpuscles. These Pacinian corpuscles are also known as laminated corpuscles or laminated receptors. So Meissner's corpuscles for touch and Pacinian corpuscles or laminated corpuscles for pressure and deep vibration. Ruffini corpuscles these are the capsulated or encapsulated receptors, the spatial sensory cells, these cells, which are neuroepithelial cells and modified as a receptor, are seen in case of the smell, that is in nose mucosa, nasal mucosa, in retina, and in the internal ear for hearing and balancing. So these are spatial sensory cells. One other type of receptor known as genital receptor, also known as Golgi-Mazzoni receptors. These receptors are for fine touch on the genitalia. So receptors are non-encapsulated receptors in form of free nerve endings, root hair plexus around the hairs of the skin, and the Merkel's disc or Merkel's corpuscles in the non-hairy part of the skin, that is the palm and sole. On the other hand, there are encapsulated or capsulated receptors which are seen in form of Meissner's corpuscles for touch, Pacinian corpuscles for deep vibration and pressure, also known as laminated receptors, Ruffini corpuscles, spatial sensory cells seen in nasal mucosa, the eyes and the internal ear, and the genital receptors known as Golgi Mazzoni receptors for fine touch and other types of stimuli on the genitalia. On the basis of the type of stimuli, which is perceived by these receptors, there are different types of stimuli. One is type of stimuli, one is mechanoreceptor or mechanoreceptor. There are some stimuli which are mechanical, known as touch and pressure. There may be chemoreceptor, touch, pressure and vibration also, and the hair cells of the internal ear. The chemoreceptors, these are the special chemicals which are received by the receptors in case of the smell, that is olfaction in nasal mucosa, and in form of taste on the tongue, taste receptors, and in carotid body. So these receptors are seen on the olfactory mucosa for smell, taste on tongue and carotid body. The other type of the receptor on the basis of type of stimuli are the photoreceptors. The well-known organ of the body which receive the photosensation are the eyes, retina, so vision in form of the photoreceptor, the thermoreceptor, in the form of temperature, either cold or hot. So these are thermoreceptors, and the nociceptor in the form of pain and the temperature. So on the basis of type of stimuli, there are the mechanoreceptors, touch, pressure, vibration, hair cells. The chemoreceptor in form of smell, taste and carotid body, in form of photoreceptors, that is sense of vision, from the eyes and in form of thermoreceptors for hot and cold temperature, and in form of nociceptors for pain and temperature. There is already a long lecture on neuronal cells, that is neurons and nervous system. Now there is the other system in the CNS and outside CNS included in the nervous system, that is non-neuronal cells or non-neuronal tissue, or also known as glial cells. Glial cells, also known as neuroglial cells, the cells which are helping for the neuronal cells provide scaffolding for the development and growth of the nervous system, help in maintaining the internal environment or internal milieu of the brain and spinal cord to act or to function the brain and spinal cord in the favorable environment. These cells are known as glial cells. There is a little review of the development of the central nervous system. Hopefully you all know there is a neural tube which is having anterior and posterior neuropore, closes on 25th day and closes on 28th day. When I make a section from here, there will be initially a single layer of cuboidal cells or low columnar cells resting on a basement membrane and having a cavity. This cavity will form the future ventricular system. These low columnar cells or cuboidal cells are initially single layered, but as time passes by, this single layered epithelium become multi-layered and it will divide mitotically and will give origin two types of cells, spongioblasts and neuroblasts. The cells migrating outside in the form of spongioblasts and in form of neuroblasts. Spongioblasts will divide mitotically and will give origin to two types of cells. One is astroblast and other is oligodendroblast. These astroblasts will mature and form the asterocytes. And these oligodendroblasts will give origin to the oligodendrocytes. Or also known as oligodendroglia. Oligodendroglia. So in the neural tube, the spongioblasts will mature and form two types of cells. One is oligodendrocytes or oligodendroglia and other is the asterocytes or the astroglia. These cells, which are astrocytes, will form two types of cells. One is protoplasmic which are seen in gray matter. That is the just outer layer. That is, this is ependymal, and then that is mental zone of the neural tube. So these cells will remain inside the mental zone and or intermediate zone and known as protoplasmic astrocytes. And second type is the fibrous or fibrillary astrocytes, which are seen in the white matter. These types of astrocytes will migrate outside the mental zone and will enter in the marginal zone. There are 5 to 10 times in number. The glial cells are 5 to 10 times to the neurons in number, but form only half volume of the neurons. So there are two types of astrocytes. One is the protoplasmic and other is fibrous. This fibrous type of astrocyte is having multiple processes. Both are having multiple processes. Cell is small, having multiple processes which are thicker in case of protoplasmic, and processes are thinner in case of fibrous or fibrillary type of the astrocyte. So here is protoplasmic astrocyte, which are seen in the gray matter or initially in the mental zone or intermediate zone of the neural tube. On the other hand, the fibrillary type or fibrous type of the astrocytes are having small cell, having processes which are very thread-like and thin. These are multiple. These cells provide the scaffolding for the development of the CNS, and these cells also provide the nutrition to the neurons or adjacent neurons. Here is a cytophysiological unit. In my lecture on oogenesis, I have already told what is a cytophysiological unit. When one cell enlarges, this nucleus remains small and size of cell enlarges very much, as seen in case of oogenesis, where secondary oocyte and primary oocyte enlarge very much, and the surrounding cells known as the follicular cells in case of oogenesis. These cells which are surrounding the secondary oocyte will provide the nutrition to the growing secondary oocyte, which becomes very much in the size, and these cells with this secondary oocyte is known as the cytophysiological unit. Cytophysiological unit. And in case of central nervous system, when one neuron enlarges in size, these surrounding astrocytes will surround the neuron and provide the nutrition and scaffolding. These neurons in the CNS which are secreting the glutamate. One neuron is having 50 to 10 surrounding 50 to 100 surrounding astrocytes. So surrounding astrocyte with neurons are known as the cytophysiological unit, as seen in case of oogenesis. Besides these, protoplasmic and fibrillary or the fibrous astrocytes also maintain the concentration of extracellular potassium, which is increased in amount during the excitement of the brain and spinal cord. Besides these functions, this is also having a very important and a specific function, that is forming blood-brain barrier. Blood-brain barrier. The separate lecture on the blood supply of the CNS will be dealt. During that lecture, it will be dealt in detail, but the brain is having a barrier to allow the specific metabolites and the ions or the toxins to enter inside the parenchyma of the brain. So the astrocytes form a barrier known as blood-brain barrier. The capillaries, all capillaries of the brain are very much in this blood-brain barrier and form about a area of a golf court. When these all capillaries are separated from brain and form a network which will be the size of the golf court. So brain is very rich in capillaries, but these capillaries are not fenestrated or not having the pores like in the systematic circulation. So capillaries which are in the brain are not fenestrated or not having pores. These capillaries are having the endothelial layer and the basement membrane. Outside this basement membrane, these astrocytes will form blood-brain barrier by sending the processes, and these processes will form the foot plate, end foot plate or end feet plate of the astrocyte. Here is other foot. This feet, a continuation of layer of these end feet of these astrocytes will help in forming the blood-brain barrier. On the capillary level, it is formed by the endothelium, the basement membrane and end feet of the astrocytes. These barriers will selectively allow the toxins, the ions and the metabolites to enter inside the brain parenchyma. The other type of the glial cell is oligodendrocyte or oligodendroglia. As you have already seen, the myelination of the peripheral nervous system, the oligodendroglia. Oligo means scanty